Hi everyone. Today let's discuss the thyroid gland's physiology. And in this video, I'm going to focus mostly and mainly on biosynthesis and storage of thyroid hormone. I'll try to explain it as easy as possible. Okay? This thyroid gland contains follicular cells, colloid, parafollicular cells, capillaries. Okay? In these, the follicular cells gives rise to certain hormones and parafollicular cells gives rise to certain hormones. The follicular cells gives rise to T3 and T4 and the parafollicular cells gives rise to calcitonin. Sorry for the handwriting. And then let's see a crisp intro for thyroid hormones. There are two principal hormones. The one is thyroxine and the other is triiodothyronine. Thyroxine is also called as T4 and we call triiodothyronine as T3. The word itself has tri, so three. Okay. The thyroid output on the whole consists 90% of thyroxine and just 10% of triiodothyronine. So when we see that, we would generally think that the least secreted hormone will have the least action, but it is not. Okay. The thing is that this 10% of T3 has most of the thyroid actions of thyroid hormones. Okay. And there are two other hormones also, and they are reverse triiodothyronine. We also call it as reverse T3 or T3, and then calcitonin. I have already said that calcitonin is secreted by parafollicular cells and not follicular cells. Okay, calcitonin deals with the word itself has calci, so calcium homeostasis. And then reverse triiodothyronine. It just constitutes less than 1% of thyroid output and it is an biologically inactive thyroxine. It is not an active hormone. Okay. Now let's see biosynthesis and storage of thyroid hormones with this picture. I'll try to explain it as easy as possible. Do focus. Okay. Now we are going to see how the iodine that we are taking in our food is going to form or is going to influence the formation of our thyroid hormones, that is T3 and T4. Okay, let's see. The iodine that you're taking in your food is not going to enter your capillaries in the form of iodine. It is going to enter in the form of iodide. Now it would be more easy. Okay, iodide, but in your exam hall, everything will forget. You will be confused like it is iodide or just iodine. Remind that it enters as iodide and inside it gets converted as iodine. Okay, iodide is taken up by these follicular cells not in a passive way, but in an active way. What is active transport? It requires energy and it requires a transporter, and the transporter here is NIS transporter. The abbreviation for NIS is sodium and iodine symporter. What is a symporter? Symporter is nothing but the one or the transporter which transport two different molecules but in same direction. And the direction here is from the capillaries towards the follicular cells and never in this way. Okay. Now this iodide has entered into the follicular cells. The step one is over. Step two is that there is a molecule called as thyroglobulin, which is nothing but, let me write it down with abbreviated form, thyroglobulin. It is nothing but a large glycoprotein which is present on the surface of the rough endoplasmic reticulum. And this thyroglobulin is not secreted in a protein-like form. It is secreted as individual peptide units. And this peptide units, they get secreted in this follicular cell near the basal membrane and they move towards the apical membrane. It enters the lumen of the follicle by exocytosis, and this exocytosis is assisted by an enzyme called as peroxidase. very important enzyme. So now the thyroglobulin has entered the lumen of the follicles. We have to bring iodine also inside. Sorry, iodide also inside. Okay. Now this iodide travels from the basal membrane towards the apical membrane easily. But to enter the follicle's lumen, it requires a transporter called as pendrin. Never ever forget the name of this transporter, because in further we will see another transporter called as megalin, which you will confuse in the exam hall. You would have heard something as IP address. I remind it in this way. I forget about this. I will remind that name somehow. So iodide pendrin. Okay. Once this iodide enters the lumen of the follicle, there is one enzyme who waits eagerly, which is peroxidase. What he does is that this enzyme converts iodide into iodine again. Now there is going to be iodination of this thyroglobulin. We may think how this thyroglobulin is going to get iodinated. What are all the molecules present in thyroglobulin? Chill out. There is just going to be nearly 140 residues of tyrosine molecules. What is it? Tyrosine molecules in the thyroglobulin. And this tyrosine is going to serve as a substrate for this iodine for the iodination process. Right? You're clear? Now let's see how this iodination takes place and how this T3 and T4 are formed in another picture. This is the tyrosine molecule. And when this iodine molecule reacts with it, it iodinates with this tyrosine molecule in two different positions. The first position is number 3 and the second position is number 5. 1, 2, 3, 4, 5, 6. This is the numbering pattern. Okay. When it gets iodinated in the third position, it forms a compound called as monoiodotyrosine. When it gets iodinated in fifth position also, it forms diiodotyrosine. There are two iodine molecules, right? See, and now let's see how T3 and T4 are formed. It is a simple calculation. You have numbers 1 and number 2 with you. Mono means 1 iodine, di means 2 iodine. For T3, you need 1 plus 2. For T4, for the number 4, you need 2 plus 2. 2 plus 2, 4. 1 plus 2 is 3. So you need MIT and DIT to form T3. You need DIT and DIT to form T4. That's it. So now there is a colloid which is formed, which contains the thyroglobulin with attached GIT, MIT, T3, T4. This colloid remains in the follicular lumen for several months, like 1 to 3 months straight. 1 to 3 months it remains in this lumen itself. When there is a necessity, this exits into the follicular cell and then the T3 and T4 are released. Now let's see how it exits. Well, how did thyroglobulin enter? It entered through exocytosis, right? Now it is going to exit by endocytosis, and that too by pinocytosis. Okay? Here, as I already said, there is a transporter called as megalin transporter, which is a thyroglobulin exit transporter. Okay, now this transporter is going to help the transport of this colloid into the follicular cell. A pseudopodia is formed and this colloid is transported just as the colloid droplet into the follicular cells. The colloid droplets react with this lysosomes. What does lysosomes contain? They definitely contain proteases, right? So this protease reacts with the thyroglobulin, which is present in the colloid, and it helps in the release of DIT, MIT, T3 and T4 into the follicular cell. Okay, everyone are released DIT, MIT, T3, T4. This T3 and T4 will enter the capillary just by a passive process called as diffusion. There are some capillary plexus which is present, and with the help of capillary plexus and by the help of diffusion process, this T3 and T4 diffuse out into the capillaries. The fate of DIT and MIT, an enzyme called as deiodinase, it readily waits for the release of this DID and MIT from this colloid. So once it gets released, this deiodinase converts them back into iodide and tyrosine residues. So these two are reversed and they are used again. They are recycled. Okay. So the fate is that DIT and MIT are converted into iodide and tyrosine and reused, and T3 and T4 are released into the capillaries. Do like, share, and subscribe the video. Thank you for watching the video. Do comment out if you want any specific topic to get discussed.